and broadcast of Science Matters. We've got two studies today. The topic is long COVID, a very serious chronic problem for millions of people. We know that 7 million people have died approximately, so millions more have this. And as yet, no defined cause. So two recent studies have come a long way to tackling this difficult mystery, long COVID. So the first study is core mitochondrial genes are downregulating during SARS-CoV-2. That's true in rodents and human hosts, at least according to the study. The study is giving compelling evidence that long COVID is not infused with large amounts of hypochondria. Don't worry, Dr. Gonshor is going to explain, but rather an attack directed at mitochondria throughout the body's organs.
I thought that was very funny. I actually thought that was great.
So the study is hot off the press. It does a very detailed metabolomics investigation with some unusual findings and is titled Serotonin Reduction in Post-Acute Sequelae of Viral Infection. Very interesting. The two are together for a reason, which will all become very clear. Dr. Gonshor, please take it away.
Well, thank you, Reed. Well, as you know, we always say that in each of these sessions, we are taking a deep dive into recent research, sometimes literally very, very recent research, that brings to light objective scientific evidence on the topic at hand. And today, we will be highlighting the results of two very recent studies that are beginning to show the confluence of mitochondrial dysfunction, interferon action, which is part of the immune system, and a central role being played by the vagus nerve. So let's look at the actual evidence, because science matters.
Well, thanks again. And before we go on with these amazing studies, Dr. Gonshor, last week in our discussion of sea gas sting, how it drives age-related inflammation and neurodegeneration, and that led to people sending some questions, such as the following. Can you discuss the concept of inflammaging?
Well, inflammaging or inflammaging is a term that started to get used several years ago because we began to see so many people suffering from this, maybe because we have an aging population, but chronic low-grade inflammation has really begun to receive considerable attention, with studies suggesting that it may play a causal role in dementia, in late life cognitive decline, and a number of other age-related situations. Inflammation is a ubiquitous feature of aging that can occur secondary to a whole number of things, physiological and disease states, which include things like obesity, which of course is an epidemic on the North American continent, cardiovascular disease, and of course infection. And especially among older adults, chronic inflammation can also occur independent of disease as a result of several age-related physiological changes. And what are they? Well, one of them is cellular senescence. In other words, the cell begins to slow down its function. And with that, there's the accumulation of cellular debris. And the body knows it's got to get rid of that debris. And in doing that, it can sometimes inadvertently, in effect, destroy the cell. That isn't a problem if it's just a few cells, but if that starts to become a lot of cells in the body, you end up with trouble. And so it's this age-related predisposition for chronic inflammation that has coined the term inflammaging. Systemic inflammatory signaling, which is characterized by the proliferation of pro-inflammatory mediators, that is, mediators that create inflammation throughout the body, that can directly influence the functioning of all parts of the body, including the central nervous system, the brain. And because of that, it can cause changes in cognition and in general behavior. So this systemic inflammation can both incite and perpetuate a pro-inflammatory response. That means increased inflammation within the brain, neuroinflammation. And it can promote a number of other molecular changes, such as, tragically, beta amyloid oligomerization, which is the hallmark of people with Alzheimer's disease, that and tau phosphorylation. So that happens. So, which ultimately, when that happens, it leads to pathogenic changes in brain structure and function. And it will lead, as I said a moment ago, to Alzheimer's and Parkinson's, which is a major scourge in our society and getting more and more prevalent as we age.
Yeah, thanks for that. It's just fascinating how much metabolic chaos exists,
Yeah,
as we call
right.
it. So thanks for everyone for submitting your questions. They're very important to us all as we learn together. Thanks, Dr. John Gonshor. Now let's jump into today's two studies.
All right, let's do that. Okay, so I have to say something. I was originally going to speak about number one. That was going to be the only thing. And then literally a study came out just days ago about a second very interesting way of looking at what SARS-CoV-2 is able to do. And it was because of that I changed the way we were going to approach this and added a second study. So I think we're serious when we say that we're trying to stay on top of very recent studies. And this is a classic example. So with that as a bit of a anecdote, let's take a look at this and see what's going on.
Wonderful.
So we're going to look at core mitochondrial genes and how they're downregulated, meaning that there's less of those genes working during SARS-CoV-2, that infection, which we're all aware of, both in rodent, mice, rats, and human hosts. And the second study is serotonin reduction in post-acute sequelae of viral infection. And we'll describe what that means. So here are the two studies. And as you know, we put that on a site where you can go to it if you'd like to read the studies. And one of the conclusions of the first study, and here is in quotes, this is what the authors are saying, is when the viral titer first peaks, that's when you first have the COVID infection, there is a systemic host response, and then it's followed by a viral suppression of mitochondrial gene transcription and induction of glycolysis. And we're going to go over that in a minute, leading to the deployment of antiviral immune defenses. We're going to describe what all of that means in just a minute. And the second study, which is very seductive, says that post-acute sequelae, that means untoward effects, things that you weren't expecting, post-acute sequelae of COVID-19, which they're putting as an acronym PASC, that's in effect long COVID, is associated with, of all things, serotonin reduction. So what are we talking about here? Well, let's take a look at the first study. We know, or we think we know, that the mechanism that SARS-CoV-2 uses to create this negative long-term effect on cells, compared with other coronaviruses, up until now, it's remained elusive. That mechanism has been elusive. We really don't know what this long COVID business is all about. And there's evidence now that's suggesting that mitochondrial genes, remember the mitochondria in the cells are the energy creating aspect of the cells. Well, the genes that control mitochondrial function can be negatively impacted by the virus, leading, because of that, to dysfunction in a whole number of organs beyond the lungs, which, if you remember, when COVID started, that was the devastating part, was the effect on the lungs. So what does the study do? They analyzed mitochondrial gene expression, was it up or was it down, in both nasopharyngeal and autopsy tissues from affected patients, as well as in animal models. And what were they looking for? They wanted to detect if there were any differences from normal to people and animals that were involved in a COVID infection. And what are the findings? Well, one of the findings is that if you look at nasopharyngeal samples, that is samples taken from the nasal area and the upper respiratory tract, they found that the virus, and I put this in quotes, blocked the transcription of a subset of nuclear DNA encoded mitochondrial OXPHOS genes. What does that mean? We know that normally a cell has mitochondria that use oxidative phosphorylation. It's a way of creating a tremendous amount of energy that the cell normally needs. But what happens, it seems that when you get this virus, it stopped the normal oxidative phosphorylation and went to a much less active way of producing energy, which is glycolysis. So when and this is a metabolic issue. Here's metabolic chaos again. If you go from oxidative phosphorylation to glycolysis, that ain't good. And so when that happens, it activates the host immune defenses, including an integrated stress response, because the body says this isn't good. So when you look at autopsy tissues, what you find, interestingly enough, is that the mitochondrial gene expression, that is this change that takes place when you first get COVID, what you see is that it recovers. Those cells in the lungs recover, but the mitochondrial function remains suppressed in other organs. The lungs did fine. They had a problem in the beginning, and in some people it was a major problem, but it recovers. And yet in the heart, the kidneys, the liver, that gene expression for the mitochondria of those cells in those organs remains suppressed. So what they're saying is the host cells respond to initial infection in a way that involves the lungs, because that's where COVID hit in the beginning. But over time, the mitochondrial function in the lungs is restored. While in the other organs, in a very perverse way, by the way, particularly the heart, that mitochondrial function remains impaired. Really very strange. And so the study is providing very strong evidence that we need to stop looking at COVID-19 as strictly an upper respiratory disease or even a lower respiratory disease in the lung area, and start viewing it as a systemic disorder that impacts multiple organs, and because of that leads to something which we're calling long COVID. The continued dysfunction, which is observed in organs other than the lungs, suggests that mitochondrial dysfunction could be causing long-term damage to the internal organs of these unfortunate patients who are suffering from what we're calling long COVID. So a potential therapeutic target could be microRNA 2392, which is shown to regulate mitochondrial function in human tissue samples used in the study. This microRNA is upregulated in the blood of patients infected with SARS-CoV-2. That means upregulation means it increases. So this microRNA is increasing in the blood of the patients with COVID-2, not that something you'd normally expect. So if you neutralize this microRNA, it might impede viral replication, and it could provide an additional therapeutic option for patients at risk for more serious complications that are related to the disease. So what are the key takeaways of study number one? Number one, mitochondrial genes can be negatively impacted by SARS-CoV-2, leading to what? To dysfunction in multiple organs beyond the lungs. And COVID-19, therefore, is not strictly an upper respiratory disease, but rather a systemic disorder that is impacting multiple organs. And if you can neutralize one of the things that is causing this, in this case, microRNA 2392, you can provide an additional therapeutic option for patients at risk for more serious complications. So that was very interesting, and that would have been sufficient for the lecture, but then along comes this study about serotonin reduction in long COVID. And what do they say? Post-viral syndromes associated with serotonin reduction, which may contribute to the neurological and cognitive symptoms that are seen in individuals with long COVID. So what they're saying is serotonin and its reduction in function may be playing a role in what people are having as signs and symptoms when they say, I have long COVID. What does the study do? Well, what the study did, it's a metabolomic study. It studies metabolite signatures that are associated with long COVID. It focused specifically on metabolites whose concentrations are perturbed. They're changed, in some cases negatively, both in acute COVID-19 and in patients with PASC. Remember, that's post-acute sequelae of COVID-19. Another term for that, long COVID. So they're focusing on the metabolites of people with acute COVID-19 and those unfortunate people who have long COVID. And among the metabolites measured, the molecule that seems to be most significantly associated with long COVID, of all things, is serotonin. And we're going to go over that in a minute. So viral, inflammation-driven serotonin depletion can be caused by what? Well, by the reduction of tryptophan absorption. We'll go over that in a moment. Thrombocytopenia. Again, we'll go over it in a moment. And increased MAO expression, meaning increased amounts of MAO. And what is MAO? That's monoamine oxidase. What is monoamine oxidase? That's the enzyme that removes dopamine from the brain. And so all of this is causing, amongst other things, an increase in MAO, and therefore it's removing dopamine from the brain more quickly. So the presence of viral RNA and then downstream interferon responses, and interferon is a very important part of the body's immune response, causes a decrease in serotonin. And the mechanisms that it uses is it diminishes the uptake of serotonin from its precursor tryptophan in the GI tract. It reduces the storage of serotonin in platelets, because that's where it normally gets stored, due to thrombocytopenia, which is a big word for a deficiency of platelets. And it enhances the turnover by what? By MAO, which is the serone metabolizing enzyme. So the important consequence of peripheral serotonin deficiency, what is that? A reduced activity of the vagus nerve. There's another component, which is very interesting, which in turn is going to do what? If you decrease vagus nerve function to the brain, it's associated with hippocampal dysfunction and memory loss because of that. All the things that these people with long COVID are talking about. So here's a representative graphic. So what does it show? There's a viral infection. Viral RNA infects the cell, and serotonin is depleted by virally induced type 1 interferons. So interferons will begin to do what? They're going to begin to reduce serotonin from being produced due to a change in the gastrointestinal cells of the intestine which normally take tryptophan, which we have in our diet, and that tryptophan is turned into serotonin by one of the enterochromatophin cells of the GI tract. And guess what? It diminishes both uptake of tryptophan and its ability to create serotonin. And if that wasn't bad enough, it creates hypercoagulability. So the serotonin stays in the platelets and doesn't get out into the blood, where it can then move to where it needs to in the brain. And on top of everything else, the vagus nerve has an effect on the brain, so that peripheral serotonin deficiency impairs cognition. How? Via a reduction in vagal signaling, which is coming due to a reduction in serotonin. And so all of that causes a real problem. And so you're saying to yourself, how can you target certain things in terms of clinical interventions to try to prevent and treat long COVID? Well, serotonin levels, if that is part of a major part of the reason for long COVID, serotonin levels can be restored and memory impairment reversed by doing what? By creating precursor supplementation. In other words, taking supplements that are going to be like SSRIs. What are SSRIs? Most of you know that's for selective serotonin reuptake inhibitors. You inhibit the destruction of serotonin in the brain so it sticks around longer. And we know that that class of medications, which are given massively to people with these problems, these medications are most commonly prescribed to treat depressions. Well, depression. Well, guess what? It could be one of the things used for this particular problem. So this study, together with recent findings that link depression with cognitive impairment in long COVID, and the effect that we know of SSRIs on vagus nerve activity, call for the assessment, a real assessment of targeting serotonin signaling for the prevention or treatment of neurocognitive manifestations. It may very well be, therefore, that we need to look at targeting serotonin replenishment and to make sure that it is not destroyed by the SSRIs in order to be able to have a way of dealing with long COVID. So what are the key takeaways? Long COVID is associated with reduced circulating serotonin levels. Number two, serotonin depletion is driven by viral RNA-induced type 1 interferons. And you're going to hear about interferons again and again. It's an extremely important part of the immune system. And these IFNs, type 1 interferons, decrease serotonin through decreased tryptophan uptake and by hypercoagulability in the platelets. And peripheral serotonin deficiency impairs cognition via reduced vagal signaling. And so that's my very quick summary of both of these studies.
I can't thank you enough, Dr. Gonshor. This is very enlightening, and known for reducing to the ridiculous. I will say that COVID is the gift that keeps on giving. Really amazing the degeneration and breakdown once you've supposedly recovered from it.
Exactly. Yeah.
And I know that 3 million people in the United States alone died of COVID, but it had a 98.2 survival rate. If you do the math, I think it's around 90 million people probably have long COVID, or at least they have the potential for long COVID. So it puts us in a position where we can help a lot of people if we can sort this out. And so thank you again for those great studies. Folks, please subscribe, like, and share if you enjoyed this episode. Submit your questions, comments, and suggested topics to fdntraining.com/science matters, and grab a copy of the studies at fdntraining.com/studies. Join us again in 2 weeks on Science Matters when we'll talk about antioxidants stimulate BACH1 dependent tumor angiogenesis because science matters.